Yeah, my name is Harald Schmidt. I'm a medical doctor and pharmacist at the University of Maastricht.
My name is Dr. Benedikt Frank and I'm a clinical neurologist from University Clinic in Essen.
My name is Hermann Mucke. We have a small but internationally active consultancy for the life sciences, and my role in REPO-TRIAL is basically getting the science to the product level.
My name is Baumbach. I am chair of the experimental bioinformatics lab at the Technical University of Munich, and within REPO-TRIAL I'm responsible for everything that involves computation and in particular bioinformatics.
REPO-TRIAL stands for the repurposing of registered drugs for a new indication with a completely new approach. We have identified a group of diseases which are genetically linked or by comorbidities, and we redefine those diseases now by a common mechanism. We can also thereby diagnose patients not only by a symptom but also by detecting that specific disease mechanism and then treat them in a highly specific manner by repurposing drugs that target that mechanism for this new indication. This whole process requires a lot of bioinformatics input, and we then interlink with biomedical scientists and clinical scientists and regulatory experts. Our current ways of drug development are actually in a major crisis. The drugs that we have on the market are very often imprecise. They don't work for many patients. And the discovery of new drugs is actually since the 50s of the last century in a constant decline. We seem to have a conceptual problem how we discover drugs. The accelerating knowledge gain has the problem that a lot of the biomedical research is unfortunately irreproducible. Over 50% of the published data are affected by that. And secondly, a major problem is the current definitions of diseases that we use, which are mainly based on symptoms or on organs, but not on mechanisms. Most diseases are actually not understood. What's the causal mechanism? Actually, I think we need a complete new definition of disease and thereby a complete new structure of medicine in total. And REPO-TRIAL provides one important mosaic stone in this process. One of my favorite and I think easy to understand example is hypertension. 95% of patients with elevated blood pressure are diagnosed with so-called essential hypertension, which means you have elevated blood pressure but we don't know why. And these patients are then treated with a drug or with drugs that dilate blood vessels. Therefore the symptom disappears, but the cause is not treated and remains unknown. Our approach to actually define a disease by a mechanism will lead, for instance, to a completely new definition of hypertension. It's a common saying that diseases have several causes and are therefore complex. We would challenge that. We would say diseases are only complex because they are poorly defined. So coming back to my example of hypertension, I would say hypertension is not one disease. It's the symptom of several mechanisms. And what we will do is we divide now hypertension based on this mechanism into several diseases, and then they will become not so complex in the end. We apply this in the moment for stroke, where we have no registered therapy available, and we have found a drug that is used for kidney disease and a drug that is used for a heart disease. They both can be repurposed, that's the technical term, for stroke.
Brain and heart are highly connected as both share the same risk factors and disease that lead to stroke or heart failure. These are, for example, high blood pressure, high cholesterol levels, less physical activity, and diabetes. In the REPO-TRIAL project, we have the huge advantage that we investigate drugs that are already known. So we know the spectrum of effects of these drugs and thereby can minimize, nearly neutralize potential side effects of these drugs. We are performing a randomized double-blind controlled study for the stroke trial, which means that neither the patient nor the physician knows in which group the patient is. So we don't know if they are applied to the effective treatment, the study compound, or if they are applied to the placebo group. We will investigate all products on top of the standard of care, which means that the patient will get any therapy that is at present known to be effective. The patient will get the standard treatment and additionally, with an approximately 50-50 chance, the investigational product as well. We believe in the therapy that we will test during the study, and when the treatment shows to be beneficial in this trial, we will have to do another study, and then the drug potentially can be used in everyday life.
In silico means in the computer, and it started with structure-based design. Because once you have the three-dimensional structure of a target, you can build a model, and you can much easier build a model of the small molecules with which this interacts. So it started out more or less as a game between key and lock. If it fits, you can turn and it has a certain action. Now it's much more complicated. We have advanced transcriptional screening and we are using quantum chemistry and all of the cutting-edge stuff. I think it's fair to say that there's only a very small fraction of compounds that did not draw any benefit from in silico screening and modeling at some stage of their development. Results from in silico screening can be very surprising. You can get a hit on asthma for a drug that had been originally developed for treating intestinal parasites. This is just the first screen. It doesn't mean that these results actually stand up to animal trials or to the clinic. It's just going through the whole thing without having the compounds actually in hand. This is the great advantage of in silico drug repurposing. You don't need the physical compounds. You can do it all in the computer, and those systems are very diverse. We have some which would run on any desktop workstation, and others need an array of supercomputers. Well, 50 years ago, things were very different from nowadays. We knew about very few molecular targets. And on the other side, it was not very complicated to do the regulatory side. Science in pharmacology was, to a very large part, animal models or isolated organs. And if it worked on this level, you could relatively quickly go to clinical trials. This was quick, and the regulatory dossier in many cases had maybe 100 or 200 pages. No comparison to what we see nowadays. Then things changed. Molecular biology came in, which meant that we discovered molecular drug targets, G-protein coupled receptors, enzymes, transporter molecules. And now the entire thing has been turned on its head. Scientists first look at the molecular target, and they know if they inhibit this target, if they activate it, this has certain consequences in an animal model and in a disease. And this is also what regulators want, because drug targets and diseases have been categorized into specific boxes, so to say, meaning that a target has to fit in a box and has to fit with a disease. This has advantages and also drawbacks. The advantage is you have a line where you can go down with your development, and you know if your drug and your program produces results that fits this path, then you have a very clear and direct path to approval. The drawback is it hampers innovation. The developers are forced into a tunnel vision. They have, for instance, a kinase inhibitor of some type, and from all experience you know this will be good for cancer, just to give an example. But kinases are very diverse molecules and they occupy many positions in the biochemical pathway, so they can just as well be useful in a rare disease, which has nothing to do with cancer. Our current way of developing drugs tends to ignore these things.
The biggest one is the sheer size of the data. Just to give an example, the next generation of sequencers generate between 4,000 and 6,000 gigabyte of data per day per machine. This amount of data cannot be just copied over the internet anymore or with a USB stick. Raw data processing itself is already a massive challenge. Second biggest challenge is the heterogeneity and diversity of the data. Right? So mass spec data looks totally different from genetics data, that looks totally different from electronic health record data. Right? So the integration of all this data is a massive challenge because all these data sets have like different little properties and so on, and the devil's in the detail. Within REPO-TRIAL, our job here is to integrate the data such that we can extract mechanisms, meaning affected submodules and molecular pathways that then define mechanistic causes for diseases that stratify patients, the basis for what we call de novo endophenotyping. And the third challenge is data protection, data privacy. In particular, patient data is quite sensitive. It needs to stay within the hospitals. There's many national laws, but also European laws. For example, the GDPR. We need to ensure that patients have always full control over their data and that the data is staying within the hospitals. So in classical artificial intelligence, all data, in order to be learned into one model, would need to be copied into a cloud system and then analyzed there centrally, which is just not easily possible here. So we need to work on technical solutions that allow to analyze the data in a distributed fashion. So we apply federated machine learning here. In particular, here for REPO-TRIAL, what we develop can be applied to all kinds of complex diseases. And of course, it's extremely important that the approaches we develop are available to other researchers. It goes without saying that we make this publicly available for the community.
Usually, once you have completed the basic science, there are regulatory requirements, phase 1, 2 and 3 clinical development of a drug. This takes at least 10, sometimes 12 years. Our approach, of course, can build on the knowledge that we already have of the registered drugs. So the majority of our work will be moving from in silico directly to the clinic. We have identified about 50 drug candidates, probably in the end focus on 5 and use them for 2, maybe 3 clinical trials. Yeah, the whole duration of REPO-TRIAL is 5 years, and I'm very confident that in these 5 years we will conclude at least 2 clinical trials. Thereafter, there is maybe 1 confirmatory trial required, which may last a year or so, and then the clinical application may be directly there. We typically know all possible side effects of these drugs, so we can determine a very good risk-benefit profile for it, because we then combine it with a mechanistic selection of the patient. The precision of our intervention will be very high, and overall for the patient, the likelihood that they will actually benefit from participating in our trial and from this therapy will be much higher. The overall costs for a full drug development process are in the moment astronomous, around $3.8 billion. Our approach basically starts very late in the process. Plus, we can use high precision, small number clinical trials. So we try to do it with a very low budget in the range of 300,000 to 400,000 euros.